Hello viewers, Goa State Research Foundation has started with this initiative of interviewing the researchers in various fields. So today we have with us Dr. Mahesh Majik. He is a distinguished chemist and associate professor at Research Development and Innovation Directorate of Higher Education. Sir has a research experience of over 17 years in the field of organic chemistry, particularly in marine natural products and next generation nucleoside. Sir has published around 60 plus articles in Scopus Index journal and has citation over 2000 plus. So on behalf of GSRF, I welcome Dr. Mahesh Majik.
Thank you, Vandana, and thank you, GSRF, for this interesting initiative. I am very happy to be part of this initiative.
So now we'll begin with the first question. Sir, welcome once again. So, sir, can you tell us a bit about your educational background and why you chose chemistry, specifically organic chemistry field for research?
Oh, well, that's very interesting. See, after graduation from Government College of Arts, Science and Commerce, Saklim, I joined Goa University to do my master in chemistry. I learned most of the basic things about chemistry, theoretical and hands-on on chemical reactions and all. And there I got fascinated towards the field of natural product chemistry, because natural products are widely used in the drug discovery process. And hence, after my MSc, I joined National Institute of Oceanography in Goa to carry or to take experience about natural product research. So there I worked for 2 years, and with that natural product chemistry, isolation and characterization experience, I thought of continuing my research in synthesizing and preparing natural products, and that's how I joined for PhD at Goa University to work in a field of alkaloids. So this is how my chemistry career begins.
Sir, so can you tell us more about this, your research in this natural marine products and its significance in our society?
Yes, so first of all here, we should aware about what are natural products. So in chemistry, one of the part of the chemistry is the natural products. Natural products are the chemicals which are prepared by usually the organisms. It's a flora or fauna, and they produce it for their own benefits. And that chemicals are usually the natural products. However, these chemicals are produced in very trace amount, okay, by the natural sources. So it is produced by that organisms, either marine or terrestrial, for their own benefit. It is having a different benefits, like they prepare it to protect themselves from the predators, because predators should not eat. If you consider the marine sources, they are usually very soft organisms, or they cannot move fast, so predator can very easily attack them, right? So that's why they produce the chemicals. That is in trace amount. Sometimes they produce the chemical to find out their mate, or sometimes they produce the chemical to protect them from injury, because there is no doctors inside the ocean or for plants. So usually they prepare their own medicines. And this is how the natural products are explored by the chemist. Afterwards, after isolating it from the natural sources, it is explored by the chemist for the therapeutic application. And if you see nowadays in the market, usually marine natural products is having a very good markets, actually. More than, I think, at today's date, if you see 2024, the estimated cost, the market for marine natural product is around 4,000 million US dollars. And in coming 10 years from here, like 2034, if you see, it will increase to double, like 9,000 million US dollars. So that tells the importance of this natural product, in particularly marine natural products.
So, sir, now you told that the marine organism, they produce some chemicals. So, in order to do your research, how you get access to those chemicals, or what are the challenges, because they will be endangered species, right? So, what are the challenges you face to do research using this marine
Well,
organisms?
here actually, see, the access to terrestrial sources are a little bit more easier because once if you collaborate with a botanist or the people who are working in the plant sciences, you can collect that material, you can identify that, and you can start working on their metabolite content. Similar way, marine natural product, if you have to work, there are three aspects. One is collection of sample. Now, for collection of sample, really, there are lots of regulation. You cannot go and collect the sample like that. Either you have to take approval from government, and only selective authorities can collect it. Okay. So in my case, I am not. When I was in National Institute of Oceanography, I was collecting the samples. But now here, what happened? I collaborate with some scientists. They collect the samples and they give it to me. Now here, collection can be done in two ways. One is what we call the intertidal collection. That means whenever there is a low tide, okay, so the rocky area is exposed like that. So from there, one can collect the sample. Some of these are endangered species. You cannot harvest, okay. And second is by scuba diving. So just going inside under the sea, and through scuba diving, you can see how many samples, what type of samples you are there. So accordingly, one can collect it. So here you have to take the proper care, because sometimes those sample when you are collecting, if you are not using the gloves and proper way of sampling, if you are not doing it, it will infect your hand and infection can occur. So these are the challenges in collection. Okay, once you have collected it, you have to store it in a very deep freeze it, or make it like or directly put it in a solvent for extraction. Usually solvent means like how we prepare a tea. Like tea powder, we put it in water and then we boil it. So whatever content is there in that tea powder is getting extracted. So similar way, we put this particular organism, a little 1 gram or 2 gram, whatever we collect, in a solvent called methanol or ethanol. Okay, and then whatever chemical present in that particular organism are getting extracted. That is a soaking method we call. And afterwards, now here the challenge, what I said is, these chemicals are produced by organism in a very trace amount, very small amount, small quantity. So it is a complex mixture will be there and very trace amount. So there are lots of challenges in purifying these chemicals. By using a chromatographic technique, one can purify these compounds. Furthermore, what is done nowadays, if you heat this compound or if you keep this compound outside, so chances of degradation is more. So that's why many people are not choosing this area, because compounds get decomposed and spoiled and all those things happen. So we should give a dedicated time for that, and it should be continuous process like that.
Sir, so I have come across many of your articles regarding next generation nucleoside. So can you tell us something about that, significance of next generation nucleosides?
Yes. So before going to the next generation nucleoside, we can also speak something about which are the marine natural product actually I worked on, and because we will get continuity for that.
Yes.
Right. So by doing this procedure, I worked on different marine natural products on isolation and purification. This involves something like a ceramide, cyanic acid, then cerebrosides. There are lots of natural products I had isolated, and most of those are published, and even industries was also interested in collaborating with me for some of this molecule for large scale synthesis. Now what happened here, whatever I isolated was in a trace amount, and that trace amount was not sufficient to do the detailed biological studies. So that's why what chemists are doing, detailed biological studies means what? See, for example, if I get something around 0.5 milligram or 1 milligram, now that 1 milligram is not sufficient to do the drug testing. Like if you had to explore as anti-diabetics agent or anti-cancerous agent or antibiotics, so lots of biological studies are done. That 1 milligram is not sufficient. Another thing is that we cannot harvest more natural sample. right? And again, separation involve a lengthy procedure also. So that's why what chemists are doing, chemists, once they get the structure of that molecule or identity of that molecule, usually pharma industries and chemists usually preparing that molecule, what we call the synthesis of that molecule, so that they will get access to the large amount of compound. Because natural sources, there is always supply issues, and that's why most of the drugs, most of the marine natural products are having good activities, but since there is a supply issue, it has not explored to that level. So that scope still exists, and chemists now started constructing those molecules. So one thing is like you have to identify which are those molecules. In literature also we get it. Okay, so you have to know which molecules you have to synthesize it, so that after synthesizing you can keep it you can give it to the collaborator or industry or clinical trials for the different studies. So that's the use of organic chemist or synthetic chemist, and lots of research is done on in these areas. So for example, I synthesize one molecule called cyanic acid. Okay, it is isolated from the marine algae, Diganea simplex. It's an algae name. Now you have to collect something around 10 kg of algae to get 1 gram of cyanic acid. 10 kg of algae and 1 gram of cyanic acid you will get. So now due to this, because it is having it is used for the neurological disorder, that compound cyanic acid found, it is isolated from algae, and it found as a very good source or source for the drugs for the treatment of neurological disorder like epilepsy, Alzheimer disease related type of thing. So people started extracting or collecting more algae from the ocean, and then government banned collection of that algae. Now what happened if somebody has to do the research? Now they cannot take it from the natural source. So now chemists started synthesizing it. So I was also involved in synthesis of that, because more than 60 synthesis is presented. But that what I found that they are very costly methods, lengthy methods, and if I want to give it to the industry, I have to do it in a very fast way. Okay, the simplest way. So nucleoside, like how I entered nucleosides. There are also marine nucleosides. And if you see the literature, you will find that the first antiviral agent used was from the marine nucleoside, this one, drugs. Now, why marine is why people are attracting to marine, that already I had covered, because these chemicals are produced by organisms for their own benefit, and that too under special condition under the sea, where there is lots of stress and all those things. So they are very special molecules. So that's why this one is there. So nucleoside. So usually nucleoside chemistry I entered during the postdoctoral study. After doing synthetic isolation chemistry, I thought I will do nucleoside chemistry. And while taking this decision, I was thinking like I was not aware about what are how nucleosides are done by the chemist, because I learned this nucleoside in 12th standard. After that, I don't think anywhere in our MSc or BSc chemistry program there are nucleosides, all those things. But still, I took this challenge. Nucleosides are usually what we call the component responsible for construction of DNA RNAs. Everybody knows about this, right? DNA RNAs, most of the people are aware, right? And the nucleoside are usually responsible for construction of that. Now, in this nucleosides, what we are having a two part. One is the sugar part, okay, usually furanose ring. Furanose ring means oxygen heterocycle, 5-member heterocycle. So furanose ring is there, and to that nucleobase is attached. Nucleobase means usually it's the nitrogen heterocycles, purine, pyrimidine. So these are the classes of that. So that is attached to that, to the glycosidic bond, and that is a nucleoside. Now these are called first generation nucleosides, having the furanose ring, oxygen, fine. But natural products, whatever we isolated from the marine sources or some other sources, this furanose nucleoside it is there in our body also, and it is a common. But when it is used as a drug, because it is having an important property, like nucleosides can be used as anti-metabolites in drug discovery. Anti-metabolites means what? It is blocking the 1 or 2 metabolic steps of DNA synthesis of that particular disease causing organism. So this is how that replications are stopped, and that disease can be cured. So that was the theory for the drug discovery from nucleoside. But with this first generation nucleoside, what had been found that when it goes into the body, it is not stable. That glycosidic bond get cleaved. Okay, so bioavailability is less in the body. So before reaching to the target, it get decomposed. That's one problem. Second problem was that's a stability issue. Second problem was the toxicity, and due to that instability, some other component form was leading to the toxicity. So rather than furanose ring, you use the thio sulfur containing nucleoside, or carbon containing or nitrogen containing. So accordingly, we are having aza nucleoside, thionucleoside, carbon nucleoside. So these are the second generation, next generation nucleoside. They are not related to the furanose ring. And with this, what will be the advantage? It will, bioavailability can be increased, means it will reach to the target before decomposition. That was the advantage. And second was its activity can be higher and selectivity is more because of that sulfur ring. Okay, for any drugs, activity and selectivity is very important. Activity means what? Very trace amount should produce the effect. That's the activity. And selectivity means what? It should reach to the target without affecting other thing. So that's why sometimes we call for certain patients side effects is there. So that means that drug is not having selectivity. That's why that side effects are seen. So for this type of things, that this type of next generation nucleosides are having a very good potential for the treatment of several diseases. Because now, like for example, selectivity, we do something called as a computer-aided drug design. When I am engineering my molecule, I try to see by computer-aided drug design that how this particular structure will bind to the receptor in the body. Okay, and this is how I will understand the selectivity aspect of that. And if I feel that it is not binding it properly and all those things, I can change there itself before going for the synthesis of that molecule. So this is how selectivity could be. You can play with the selectivity. Okay, so that is about the toxicity and selectivity.
So, sir, I have come across one of your article in Nature's Medicine. So it was related to something GATA enzymes.
Yes.
It is a very prestigious journal which is having an impact factor of 80. So can you elaborate on that, sir?
See, every researcher is having dream to publish in Nature. So I was lucky enough to select one problem which is a part of a Nature Medicine. So this problem arises like it's a simple observation. See, for research, for doing research, observation is very important. Okay, it's not like that you have to do so many things, or you have to struggle to do research, and that's okay. But if you are a good observer, you can generate your problem, whatever problem is there, you can identify that problem and you can provide a solution to that. Now, this Nature's publication is like just a discussion based problem. So just I asked my professor like, see, there is one thing happens, like when we are very happy, we promise something like that, and when we are very sad, we make certain decisions, and we always say that is wrong. Okay, and I also because I experienced that also.
Yeah,
when we are
extreme
angry,
moods.
yeah, so your decisions are always wrong. So my intention was like to discuss with a biologist why this is happening, and then we understood that when you are very happy, or when you are very depressed, or when you are under stress, the particular enzyme is produced in the body. That's what we call the GATA enzyme. Okay, and that GATA enzyme is produced like because of that, the switch between the brain and the other cell is getting off, and that's why we had proved in our experiment that with the help of biologists that the size of the brain is getting shrink little bit. Okay, and due to that, those decisions, because it's not a normal decision because of that, because size of the brain is getting shrink, so you become abnormal like that. So that's why those decisions are always wrong and all. So now how chemistry I brought in this, like why not to inhibit that GATA enzyme? Suppose GATA enzymes is produced in the body, you inhibit it. Now once you inhibit it, that means what will happen? The brain size will remain normal like that. So that's why I design a molecule, natural inspired molecule, natural plug inspired molecule, and that molecule we tested on the patient, and we found that mentally disordered patient, usually it is called MDD, mental disorder diseases. So we tested on them, and that was in collaboration with US universities, Seoul National University, and I was from Goa University that time. So we collaborated, and then we found a solution that this could be used as a drug for mental disorder people like that. So it's like, I designed that molecule from the natural product, and then I took help from the biologist and doctors from medicines and chemist, and so many people was involved. So that's why it's not like if you have to do something, very good impact research, there is always need of, either looking into interdisciplinary approach or a holistic approach, usually we call, so that you can see that particular problem from all the view, and its output is always good. So that was my work in nature medicine, like that. Yeah.
So my next question is, what are your current research or currently what you are working on in research, or what publications you are working on?
Okay. So my main areas I am targeting, like as I said, marine natural products, nucleosides, modified second generation nucleosides. However, now I am interested in exploring new natural products for treatment of various diseases. So that is like whatever is practiced by the local practitioners in Goa. So those what we call the Gauti workers or local medicines. From there, I am trying to collect the samples and trying to explore the chemical content of that for the drug discovery process. So that is related to the nucleoside conjugate, new field. So I am combining natural products, nucleoside, and preparing the hybrid. So that it will be having, as I said, in drug discovery, this type of things will give you more what we call the activity, good activity. So very little amount could be sufficient. So there will be no problem of toxicities and all. So people worried to take tablet because it is in a gram level or 500 milligram or like that. So if it is in a nanogram or milligram level, people will not worry. So that's what we call the activity, increase the activity like that. Okay, so I'm working on those areas. So one is on exploration of or method development for the new natural products from the natural sources from the Goa, marine sources, and nucleoside marine natural product conjugate for the drug discovery. So it is having really a very good future. That's what my future plan for doing the research.
So any advice or any guidance you want to give to the new researchers who will like to enter exploring this field of marine products or next generation nucleoside?
Yes.
Or drug discovery?
Definitely, I would like to say something about this because based on my research experience, what I feel is that you should keep yourself, very curious and try to keep learning every time. Research is tough, no doubt about that, but what we call dedication and focus is very important key for that. Okay, and exploring new area, because that's what is required. We have to take a chance, risk, because exploring new areas will give you new results and good results. It's not like that. See, most of the time, if you try to search something when there is a light, I think others might have already got that because there is a light, so they will get it. But if you are search something in the dark, others might have not seen that. So that's how I am exploring my research. So you try to explore research areas which people had not covered it, or try to do for the interdisciplinary this one. So networking and collaboration is very important for research also, because this will give you new exposure, and that's why I'm suggesting the young researchers to keep yourself update about research and be curious about certain things and start working on it.
Thank you, sir, for sharing your expertise and insight on this topic. It was a lovely interaction. Sir, your dedication and passion towards this organic chemistry is tremendous. So thank you, sir.
I hope this particular discussion will be useful and will motivate most of you young researchers and to take or undertake a new research areas and explore the research, not only in chemical sciences but also in different disciplines, and also it will be motivating you to collaborate and do the interdisciplinary research. Thank you one and all.